There are four basic categories of organelles. Those in charge of protein synthesis, which we will spend an entire unit on. That is the nucleus, nucleolus, and ribosomes. Also partially rough ER, but we're going to put rough ER in storage and transport. Because once those proteins are made, they are modified, shipped off to Golgi, and Golgi then sends them off in vesicles to wherever they need to go, whether that is the membrane or somewhere else in the cell. Vacuoles, again, just kind of storage of water wastes. And then lysosomes, which are basically just like vacuoles and vesicles. These are all vesicles, okay? But vacuole is a specialized vesicle for storage of water and waste, especially water in a plant cell. Lysosomes are storage of enzymes, digestive enzymes. So if it stores digestive enzymes, it's basically a stomach. It digests things. And then there are the energy organelles, two of them, mitochondria and chloroplasts. Again, mitochondria are in all eukaryotic cells, plants and animals. Chloroplasts are only in plants. Why plants are green. Another way to remember that. And then the last category is for structure and movement. We're going to talk about the cell membrane in quite a bit of detail in the next unit. But then again, cell wall for bacteria and plant cells. Cilia, which you saw in the notes, are also part of the cells that are lining our trachea. So those cilia are not moving any of your cells, but instead moving particles, dirt, dust, and mucus out of your lungs. Flagella, which again are the long whip-like structures. An example would be on a sperm cell. Centrioles, moving the DNA. Cytoskeleton, which is just like your skeleton, but just made of different stuff. Okay, it provides inner support structure. And then the cytoplasm. Objective 8 is talking, I think you have on your notes a missing title somewhere. It might be in number 7 or number 6 here. So you might want to check out your notes and just, it says on your notes objective with a dash, so you might just want to fill that in really quick. And then on your notes I combined objective 7 and objective 8 on one page so that you were printing out 3 pages and not 3 pages plus one slide, which would have been 4 pages. So you may want to separate on your notes. Just put a line underneath the structure and movement organelles. The next set is for objective 8, organelles and homeostasis, which you have all typed out completely on the list of objectives. But guys, homeostasis is just the cell's ability to maintain a balance between inside and outside, and we're going to focus a lot more of this when we get to talk about the cell membrane in the next unit. But basically the cell membrane is in charge of maintaining balance of particles, molecules moving in and out. Alright, so here's a picture of an animal cell. This is going to be really important for the test. You're going to see a picture, you're going to have to locate, they're already labeled, you'll just have to fill in the labels, but not only are you going to have to locate or identify them by their structure or where they are located, you'll also have to know what they do based on the label. So for instance, a label could be pointing to these guys right here, and you'll have to recognize them as centrioles. Basically they look like churros, those things you get like at a carnival. Okay, they're always found in pairs, and these are the guys that they kind of look like tubes, right? So these are made of microtubules, and these are the guys that are in charge of moving DNA during cell division. The mitochondria, they have this kind of bean shape, and on the inside are highly folded. This is for chemical reactions that we'll get to later this year. There's another one here, another one over here. The nucleus, right here in the middle, you can see it has those pores so that things can move in and out, but not DNA. DNA is stuck in here. Or chromatin, that's one of your vocab words, which chromatin we're just going to say is DNA. It's technically DNA in proteins, but DNA, chromatin, it's inside the nucleus. We're just going to refer to them as basically the same thing. And then inside the nucleus there, this little blob, is the nucleolus. That's where the ribosomes are made. Ribosomes are making proteins. These ribosomes are out here, these little dots. Okay, these ribosomes are making proteins. Probably the second to the DNA, to the nucleus, second most important organelle, even though they're just teeny tiny specks. We'll spend a whole entire unit talking about these guys and how they work. But the ribosomes are located on the rough ER. Here's rough ER here on the right side, and the smooth ER is here on the left. You can see the smooth ER does not have ribosomes. Why it's called smooth, because it literally looks smooth, whereas the rough ER looks kind of like rough, like sandpaper. Okay, it has these little grains on them. Smooth ER makes lipids. Rough ER makes or modifies proteins. Proteins are made at the ribosome. They are modified at the rough ER. Both of these, smooth and rough ER, are shipping their contents, whatever they're making, lipids or modifying proteins, off to the Golgi, which is this orange and red guy. Alright, they're sending those vesicles with lipids or proteins in there, or some other things, to the Golgi, which is going to sort them out, package them up into a vesicle again, and then send them off to where they need to go. So you can see here are some vesicles, and there's not much of a difference in a picture, in a book or notes or anything, between smooth ER and Golgi. They both just look like folded up tubes. That's exactly what they are. The only difference that you should notice is that at the Golgi you'll always see the vesicles right nearby. Okay, so one way to distinguish between the two. The other way is that smooth ER and rough ER are literally attached to the nucleus. You can see how the nuclear membrane kind of folds out and just becomes endoplasmic reticulum. Both smooth and rough are attached to the nucleus. And then also located all throughout the cell, it's impossible to really show this accurately because they're just everywhere, are the tubules, microtubules, and microfilaments that make up the cytoskeleton. Here's a zoomed in picture of the cell membrane. Again, we'll talk about that in more detail in the next unit, with a protein that's spanning the membrane, allowing things in and out. And you can see there the membrane is made of those phospholipids, and they're aligned tail to tail, back to back. Other things, the cytoplasm, which is just kind of the watery jello-like substance that holds all the organelles in place and is the site of chemical reactions, of which we'll talk about later this year. And some other things on here, for instance, this you can see looks kind of just like this vesicle here. Looks very similar to this purple one over here. This purple one would be something like a lysosome, perhaps. Okay, different color, but you know, basically doing the same thing. Little vesicles that contain stuff. In this case, it's a lysosome, so we're calling it a lysosome. So it contains digestive enzymes. It's going to go break things down. And that's pretty much it on this picture. Here's a plant cell. Nice thing is there's a lot of similarities. Again, nucleus, same color even. Inside is the DNA or chromatin. The nucleolus, again, is that center mass inside the nucleus. This is where the ribosomes are made. And the ribosomes will just get sent out the nuclear pores here through the envelope, through the membrane, to go to the rough ER. And there it is. So rough ER and smooth ER, again, attached to the nuclear membrane. Rough ER looks rough, smooth ER does not. And then they're going to send their contents to the Golgi. Here's the Golgi, again, just a bunch of folded up tubes, just like the ER, folded up tubes. And here are the vesicles. So shipping their contents off to where they need to go. UPS is the Golgi. We have mitochondria again, kind of bean-shaped, folded up on the inside, chemical reactions. We have a vacuole in the plant cell. Typically, maybe besides the nucleus, the largest thing inside the plant cell is going to be a vacuole. It's always going to be full of water. We all know plants need water. Not only is this vacuole providing water for things like photosynthesis, but it's also giving the cell its structure. Think of a water balloon that is filled up, right? It gives this water balloon its full structure, filled up. Take the water out, and it's kind of like a deflated balloon with no air in it. Same thing. This vacuole is pushing up against the cell wall, giving that cell its full shape, allowing that tree, besides the cellulose in the cell wall, allowing that tree to stand up 20, 30, 40, 50 feet in the air. We also have some new organelles in this picture. In green here, you can see all the chloroplasts. Again, when we get to photosynthesis, we'll spend way more time on these guys. But these chloroplasts are in charge of photosynthesis, capturing light, doing some chemical reactions, and spitting out glucose, food. You can also see lined in here like some cytoskeleton stuff. But guys, literally to point out cytoskeleton inside of a picture of a cell is impossible because, you know, literally that cytoskeleton would be all over the place. It'd be making a huge mess. It'd be in the way, and you wouldn't be able to see much. So, why it's kind of like scattered around here. Other things to point out on here that are pretty important. The cell membrane, which is like this orange colored lining inside here. The cell membrane is inside the cell wall. Okay, cell wall is the outermost part. Here's the cell wall to another plant cell, cell wall to another plant cell. You can see it kind of has a squarish block-like shape. But things to keep in mind, the cell membrane is inside of the cell wall. Cell wall is the very outside part of a plant cell. And you can see there are holes in there for items to get in and out. But keep in mind, the plasma membrane or cell membrane is inside of the cell wall. Something easy to forget, confuse. Here's a picture of the cell membrane. We'll spend a lot more time on this, like I said, in the next unit. But again, made of phospholipids and lots of other stuff. Proteins, big purple molecules here, cholesterol. There are some proteins that have carbohydrate chains stuck to them. Okay, but just in general, get an idea of what that cell membrane looks like. And its job is just to allow things in and out, regulate what goes in and what comes out.
Can you be a robot?
I am a robot. Beep boop.